So the object of today's lecture is to really kind of give you an overview of the different model systems that you can use to study human disease, and in particular ALS. So there are a lot of different examples. And what I really want to do in this lecture is kind of set up what the advantages are of each model system, particularly focusing on worms, flies, and zebrafish, because you're going to have another lecture about the mouse. And I think through all of this, you've kind of gotten some mouse model information. And I'll talk a little bit about that, but I won't dwell on it. I really want to talk about these kind of invertebrate and vertebrate non-mammalian model systems. So we'll go through kind of the strengths and weaknesses of the different models, and then I'm going to give you an example of in each one where they overexpress the same protein SOD, and kind of look at how they did it, they made the models, and what they learned, and just kind of at the end maybe have a discussion about compare and contrast, and how these models could be relevant and what are kind of the caveats. So that's kind of the goal of today's lecture. I'm certainly not giving a comprehensive overview of all the different animal models made in ALS, because that could be a whole seminar series in itself. Okay, so as you know, and as you've probably thought about in your own work, each model system has its own advantages and disadvantages, strengths and weaknesses. So I'm just going to start with the elegance and one of the strengths is that it has a, I guess you could give this as a strength and a weakness, it has a short lifespan, so 2 to 3 weeks and a 4-day generation time, so very quick, right? You can generate a lot of animals in a very short amount of time. They breed at will, so you don't have to wait for them to breed, you don't have to order breeding animals. Large numbers and easy to manipulate. They're the first multicellular organisms to have a genome sequence, so they have very strong genetics. 302 neurons, and also so the neurons are defined, and also the connectome is defined. So the circuitry of the worm is identified. Genetic manipulation is really easy, and I'll show you an example of that as we go through the worm model. And they're very strong for doing genetic screens, chemical screens, RNAi screens. Again, because you have this huge number of animals, you can get it very easily, and they're very easy to manipulate. Some of the weaknesses are the neuroanatomy and neural circuitry is different from different invertebrates, so they don't have cortex. Only mammals have cortex. They don't have spinal cord, okay? So they don't have those structures that are going to be important to the spinal cord, such as down here a blood-brain barrier. So they lack a blood-brain barrier. They have very few glia, and they don't have microglia. And this is something that Dr. Kaspar talked about last week, the role of the non-cell autonomous role of these cells in ALS and their effect on the motor neurons. So these animals don't really have those cells. The motor neurons are GABAergic as opposed to cholinergic, so this is also a difference compared to vertebrates. So they use a different neurotransmitter, which may mean that they have other differences that could affect the disease modeling. Okay, so that's worm. Hey, Darcy, do I have to point this anywhere in particular? At the screen?
At the screen's fine.
So for C. elegans, again, you have a short lifespan, longer than a worm though, about a month. 7-day generation time, so again, pretty quick still. Again, breed at will, you can get large numbers. Again, easy to manipulate. Strong genetic tools, so flies, of course, everybody knows about the strength of the fly in terms of genetics. Small genome, easy to manipulate, all the tools are in place. And also very strong for genetic screens, much like the worm in that way. Some of the weaknesses, again, similar to worms, being invertebrates, their anatomy and their circuitry are different. Again, no cortex, no spinal cord. In this case, the motor neurons are not GABAergic. also maybe more time to develop a disease, certainly an adult onset disease like ALS. But still they have this advantage of having large numbers and easy to manipulate. So again, just like worms and flies, they breed at will. You can get large numbers, thousands of embryos at any one time, and they're easy to manipulate in terms of their embryology and also the adults. Genetics has caught up to all the other model systems now. The genome's been sequenced. We're on our 10th round of the genome being sequenced. You can make transgenics very easily. You can make knockouts. Much like flies and worms, you can do all these genetic manipulations in-house. You don't have to send them away to make a knockout, make a transgenic. It's cheap. Anybody can do it. It's just a matter of doing basic lab injections. So again, you have the ease and the cost advantage. They're a vertebrate, so you can do vertebrate genetic and chemical screens in zebrafish. Not as high throughput as invertebrates, but much higher throughput than mammals. They have vertebrate neuroanatomy, so they have a spinal cord. They don't have cortex, but they have a telecephalon, which is where the cortex is derived from. They have robustglia and microglia. They have a blood-brain barrier, and they also have behavior. And I should say, too, that the flies have a bit of behavior as well, but the behavior in zebrafish is getting more sophisticated. Weaknesses, the generation time is longer, so 3-month generation time, so again, you have to plan that into your experiments. They don't have a cortex, they're a vertebrate, but they're not mammals. So then of course you move into the mouse model, and also I guess you could say the rat models. Most people still use mouse. They're mammals, which again is closer to humans. Longer lifespan, so maybe these diseases can develop along a more normal trajectory than they can in flies and worms. Again, you can do vertebrate genetic and chemical screens, but they're a lot harder in mouse than in, for example. Of course, vertebrate neuroanatomy, and they have a cortex, so you have the mammalian neuroanatomy. They have microglia, blood-brain barrier, and also robust behavior. That's been very well characterized. Weaknesses, 21-day gestation period and a 2-month generation time. So again, you can plan your experiments accordingly. Low numbers and small litters, which is definitely a disadvantage. They're not great for genetic or large drug screens, again, because of the problem with the numbers and the cost of keeping large numbers of animals. And another thing that a lot of people don't talk about, but I think needs to be considered, is that mice are very inbred. And often when you're doing drug screens or testing drugs on an inbred population, you may see a difference that really is lost when you go to an outbred population such as humans. And I should say a strength of the zebrafish didn't put on there is that they are less inbred than mammalian models. Because zebrafish, if you inbreed them, they don't reproduce. So you're forced to keep them more outbred. Okay? So those are just some of the things to keep in mind as we go along. And other things, when you're starting out to make a disease model and your favorite model organism, there are a number of things that you need to keep in mind. And I just go through some of the main ones here. So, does your model system have the cells that are affected in the disease? So it may sound obvious, but it's something to think about. The entire system or circuit may not be present. So one question you can ask is, if you have some of the cells that are affected in the disease, but not all of them, is it still relevant to model the disease anatomy? For example, zebrafish don't have a cortex, but they have a telecephalon. Is that a good stand-in or not? What are the genetics of the disease? In a lot of cases, and ALS is a perfect example, we often don't know if the disease is caused by a toxic beta function or a loss of function. So do you knock the gene out? Do you make the transgenic overexpressing a mutant form of the gene, which we'll see today is a case in ALS that's often done. But so, going in, if you don't know, you really have to make a guess or do it both ways. How will you generate the model? This kind of ties into this, but if you have to make a transgenic, and we'll come across this as well in the discussion, if you're making a transgenic, what promoter do you use? Do you want to drive the mutant gene the way it is normally expressed, or do you want to drive it in certain cell types? If you do drive it in certain cell types, is that relevant to looking at the disease as a whole, because you're no longer modeling it in all the cells that are affected in the disease case? So that's a big one, and we'll see examples of that today as well. Is this an adult onset or an embryonic or early disease? Model organisms that have shorter lifespans than humans, this could be an issue. This is true with ALS. It's an adult onset. play to the strength of the model organism. So some of these organisms, like flies and worms, super strong for genetics, maybe not so good for looking at circuitry that may not be conserved. Certain zebrafish, great for imaging, right? So that's a strength. Some models are better for looking at cell biology. So again, if you know what your goal is in mind once you build this model, is it to look at cellular interactions, right? And you've got to have all the cells present that may be important in disease. Is it to do a drug screen on a very controlled phenotype that's just in one cell that you think is really relevant? Maybe a fly or a worm is good. So all these things you need to keep in mind before you start, so that you can end up with the right model for the questions that you're really interested in pursuing. All right, so what I'm going to do next is go through 3 different examples, and focusing mostly on the worm, the fly, the zebrafish, of an ALS model, and kind of compare and contrast how they made the models, what they learned, and in the end, if you can come to any kind of grand conclusions about what's the takeaway about what's been learned about the disease. And I'm not saying there's any right answer here at the end, so you'll have to come to we'll come to a conclusion as a group, say. Okay, so we're going to look at the phenotypes that were found and compare them across the models, and kind of ask what's conserved and what we can learn. Okay. So the first one is the Elegans model. This was published in 2009. And what they did, so when they set out to make this model, again, these are all going to be based on SOD, because a lot of these were done, a little while ago. It was the first gene that was linked to ALS, and you guys have already been exposed to all the genetics, right? You guys have genetics lecture already. So of course, there's many models that you can make now, and some have been made, but SOD models have been made in all organisms. So that's why I chose this one. So what they did in this paper was to drive the mutant human SOD, G85R, and also another mutation, pan-neuronally. Okay? So they used the worm promoter, worm synaptic revenue promoter. Okay? So this should be expressed in all neurons, so pan-neuronally. I told you that worms have very few glia and no microglia. So this is how they build this model. This is a protein that should be expressed relatively early in development, but certainly should be expressed throughout development, because this is a protein that's on and is important for synaptogenesis and synapse function. So the first thing that they did here was that they showed that there's a locomotor defect. So the non-transgenic is green, the wild-type SOD, they expressed human wild-type SOD and human G85R mutation. G85R is a common mutation that's made in animal models. It's a human point mutation. And here you can see that the non-transgenic and the wild-type have a very similar locomotor profile over days. So it starts high and it goes slow as these animals move out. And you can see here with G85R, they have very decreased relative locomotion and distance. Distance is a fraction of body length. And here you can see there's another form that they used here that affects copper binding, so it's a double mutation, but they show kind of the same thing. Here's the wild type, here's this mutant, and here's the G85R. So they are seeing a locomotor defect. And they also showed in their supplementary data that they saw thrashing effects. Like this is the thing where you touch the worm and it moves about. And they also saw that was effective in SOD. So these worms have a movement defect that is very easy to characterize during their development. This is a transgenic. So I don't know in this paper if they showed it. A lot of times they don't show how many copies. Right. 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 They did not talk about that, and that's an important point. So when you're making these transgenic models, you kind of get what you get, right? And in the mouse, in the fly paper, they do show the Western. This one they did not show the Western to talk about copy number. And that's the other thing that I think is important, is to be aware of the copy number. And if you have a model like worm, it's really easy to make transgenics. So make a couple different ones and see if you get the same phenotype, or if it's graded and dependent upon the amount of mutant protein that you're expressing. So in looking, again, this is driven pan-neuronally, so they looked and they had they have a YFP tag, and they do some experiments to show that the YFP tag form is also causing movement defects. But here they're showing neuronal aggregation. So they're looking here in the aggregations in the neurons. They went on to look at different neuronal types. So here are different pansensory, three pansensory interneuron, another pansensory neuron, an interneuron, and an unknown. And this is when these neurons are born. So they were trying to look to see if there's a correlation between the aggregation and the birth date of the neuron. Like if the neuron's around longer, does it get aggregations more or more quickly? And they did not see a correlation between that. They saw that this pansensory neuron had a lot more aggregates that were larger. And the percentage of neurons that had these inclusions was greater. But they did not see a correlation between the neuron type or the birth date of the neuron. So there was really no correlation in those two aspects. As you can see here, they're just looking at the formation of these over time. So at the larval stage 3 and early larval stage 4, you can see the buildup of these, and by late stage 4, you see a quite large amount of aggregates. So here is a biochemical analysis of insoluble SOD protein. So their supposition was that they're getting aggregates, and these were not. This protein was insoluble, forming these large aggregates. And so they looked in doing biochemistry just in the whole animal. They saw that here in the supernate or the pellet, you can see that you have a pellet that has protein in it, which you wouldn't see if it were completely soluble, like you do with the wild type. So the wild type's not soluble, or not forming aggregates in pellets, but the G85R is. And this is also true of the YFP tag. And then they did a plot to show that you get high molecular weight. In wild type, you see that the molecular weight is kind of around these sizes, but in the mutant, you see this big long spread, suggesting that these are very large aggregates that you're finding. So just another way of looking at aggregates biochemically. So they're seeing them in the animal, they're seeing pellets indicative of insoluble protein, and they're seeing, when they look at these large molecular weight bands, showing that there are these formations that are occurring. And these are occurring just, again, you're only driving the neurons, these are occurring in the neuronal population. So what they wanted to do is, because they have this way to visualize these aggregates, oh, that's on the next slide, sorry. So first they wanted to do was to look at the presynaptic puncta in these animals. So they figured if you have these large aggregates in these neurons, is it affecting the formation of the synapses and the function of these neurons? And they were seeing a locomotor defect as well. So here they're just looking at a number of different markers of presynaptic terminals. A lot of these are presynaptic vesicle markers in both wild type and G85R. And they find that they're seeing a lot less of these presynaptic puncta in these animals. And here they quantitate it just by looking and counting these puncta. You see this decrease. And here what they're showing is that if they photo bleach these puncta along the nerve cord, they find that they don't see these filling in as well after photo bleaching, which is indicative of a problem with axonal transport. That's what they're saying about this. So decreased recovery after photo bleaching. And they show here that this doesn't happen with the wild type overexpression, but it happens with the overexpression. So they're getting aggregates in these neurons. They're seeing problems with movement, aggregation in the neurons, decreased synaptic puncta, and also decreased movement within these neurons in their axons. So they also looked at the neuronal processes, and they saw a slight decrease in the neuronal processes. And they looked at this both with their GFP labeling, but also with EM. So they saw a decrease in axonal bundles, which is a little bit hard to see here, and decreased mitochondria and synaptic vesicles here. So again, this goes along with their seeing a decrease in synaptic puncta. When they look at EM, they're seeing a decrease in synaptic vesicles. This is all consistent with there being a real problem with motor neurons when you overexpress this mutant SOD. And so the other thing that they tested was that to assess synaptic function, they put the embryos in a cholinesterase inhibitor. So even though I told you that their motor neurons in worms are GABAergic, there are cholinergic interneurons that interact with those motor neurons. So in wild type worms, if you put the animal in a cholinesterase inhibitor, you get animals that are paralyzed. But in these mutants, you fail to see that. Okay? So their interpretation of this is that you have decreased cholinergic synaptic transmission because when you block it, you don't see the effect. So it's already down in the animals. So again, trying to relate this neuronal pathology with regular microscope. So what they did next was to do an RNAi screen. So this was really easy to do in worms. All you have to do is feed them bacteria expressing the RNAi constructs. Just feed it to them. So you can go through tens of thousands of RNAi constructs very easily. And so what they did was feed the worms the bacteria expressing RNAi constructs and look 3 to 6 days later for changes in the number and intensity of these neuronal aggregates. So here's just a summary of some of the proteins that they found. So they saw a large number of the hits were in this protein chaperone turnover and modification category. Not surprising if you're getting these aggregates. They're probably caused by protein. It's soluble protein building up and not being cleared in the cell. And then a whole list of categories for different types of proteins. And again, because it's a worm, you can easily go in and now genetically test some of these candidates. So they took candidates from these different classes of molecules and crossed them to their lines to see if the mutants also caused a decrease in the normal aggregates. They found that indeed that when they manipulated quality control proteins and knocked them out, they again saw a change in these aggregates. So this is a real strength of this model, is to be able to have a phenotype, a cellular phenotype that you can visualize, and go in and do a screen, a genetic screen, to try to find proteins and genes that interact and lessen or strengthen that phenotype. So to summarize the worm, this worm is that driving mutant SOG just in neurons. You see these neuronal aggregates being formed. You see movement defects. You see synaptic defects. And they found that this was linked to the aggregates because lines that had more aggregates had a stronger presynaptic defect. So they really tried to make that link. And then this ability to do large-scale RNAi screens, and then to follow it up with doing genetics and doing crosses with genes. And they screened 16,000 RNAi lines. So that's a real tool. The problem with the model is that you're only expressing mutant SOD in neurons. And they didn't see any evidence of neuronal cell death, which is something you certainly see in ALS and in some of the other animal models. So whether that's because of the lifespan problem, because there's other cells that aren't expressing the mutant form of the protein, that could be contributing to the demise of motor neurons. Can't really tell from this model. Yep.
So actually, the RNAi that targeting the chaperone is worsening the phenotype by enhancing the amount of the aggregate formation, right?
Right. So what they're doing is they're knocking out by RNAi, they're knocking out a protein, and so the aggregates get worse. So their idea is then that normally those chaperone proteins make the aggregates less robust.
But during those 16,000 RNA compounds, did you say any case actually reverse the phenotype to see the rescue?
So that would be something that makes knocking out a gene would make the aggregates be worse. So normally that protein would make the aggregates worse, right?
Yeah, but the purpose of the study is to find the genetic modifier eventually to reverse the phenotype.
Right, but this would be a genetic modifier. So if you have a protein that for an RNA, if you knock it out and the aggregates get worse, that means when you add it back, right, or overexpress it, it should make the aggregates better. So they were looking for modifiers that way, things that make it better. So if you knock out something and it makes it worse, that means normally it should make it better. Is how they did this.
The second question is that aggregate is really good without.
Well, that's something that we can talk about. I mean, I'm not sure it is, right? So you don't see it in all models, and again, having different levels of protein being expressed is a really important consideration, right? Because humans, when you have the disease, you don't have, 4-fold, 5-fold, 20-fold more protein. You have just one bad copy of the gene. So it could be a problem with overexpressing the protein. But again, they have wild type protein. They're also overexpressing wild type SOD. It doesn't give them aggregates, but that's a caveat. Good. All right, so let's go on to the Drosophila model. So in Drosophila, they used a GAL4 UAS system. So I don't know if you're familiar with that, but this is a way to target expression in certain cell types by using a two promoter driver, one GAL4 and one UAS construct. And they could target this to just motor neurons. they use human SOD, but here they also use Drosophila SOD. And here they just show the comparison of the similarities. So the black are the conserved residues, and the green are where there are mutations in the human gene. Okay, so you can see that it is pretty highly conserved, and a lot of the residues that are mutant are conserved between the two, but not all of them. Okay, so they drove both human SOD and Drosophila SOD in these animals, and again they use the G85R and also A4B. So here they are showing in their transgenic, so again transgenic overexpression, they are showing the Western blots to show the amount of protein that they're driving. So this is an antibody that recognizes both Drosophila and human SOD. So here's human, there's Drosophila. So if you're comparing these two, it's quite overexpressed. And here's the relative expression profile here. And here's an antibody that just recognizes the human. And they know that G85R runs slower, the same as in mouse. So it is overexpressed. It's probably not 20-fold, but here you can see kind of a ratio of the overexpression. One problem with this though is that some of these forms, G85R in particular, are thought to be unstable, so it may be difficult to know at any one time how much is being expressed because of the turnover rate. So that's another thing to keep in mind. It may be hard to nail down how much you really have at any one time. They did try to quantitate it. Okay, so again, they looked at here. The first thing they looked at was lifespan, and they found that driving these mutant forms, both the human and the wild type of the two mutations, did not affect lifespan in these animals. So this is like worm. Okay, it didn't affect lifespan. Again, though, you're only driving this in motor neurons. And here it's just showing how they did the experiment. They looked at, again, I put this in here just to impress upon you the numbers that you can use in these animal models, right? So they did 3 different experiments to look at survival, and each of them had, 200 to 300. So again, you get really large numbers, but showed no difference in survival when you drove the mutant form just in motor neurons. So here they're looking at movement defects, and flies expressing wild type or mutant SOD showed progressive loss of climbing when compared with Drosophila SOD controls. So here what they're finding is that the human form of SOD is actually toxic. And their thought is that because there are these differences in the coding sequence, that this could be actually acting like a mutant form of SOD. They're not that highly conserved. There are these differences. And we know that in SOD protein, there's about 100 different amino acids that can be mutated that would cause ALS. So their feeling is that the human is different enough from Drosophila that it's acting like a mutant, even though you have the data for mutation per se. So over time here you can see this is this climbing index of the Drosophila climbing up the walls of the tube. It's how they do the assay, and starting at about 14 days you can see the G85R starts to show this phenotype, and then at 21 days you start to see the wild type human show the phenotype. And then by 28 days all 3, the wild type of the 2 mutants are showing the phenotype. So they're having, again, just like with worms, they're having a movement defect. They counted the nuclei in the T1, T2 thoracic ganglia, and they did not see a difference in the number of motor nuclei. So again, you're not seeing large-scale motor neuron loss in these animals, similar to the worm model. And again, perhaps that's a lifespan issue, that these neurons aren't around long enough, right? So the climbing loss, if they're seeing this change in the behavior, may be due not to motor neuron loss, but to motor neuron dysfunction. They're concluding that. This is where the only experiment where they used the eye driver. So again, they're driving the mutant forms in the eye, and what they did then is just then dissect out the eyes and do biochemistry. And they show that you have these large molecular weight forms of the protein, and that they're suggesting that this is indicative of aggregates. So like in the worm, they think that they're seeing aggregates, large forms of this mutant protein when you drive it in neurons, at least in the eye. Okay. And that's just with G85R. Then to look at the physiology, what they did was they looked at these frequency responses when they stimulate these neurons. So flies expressing both human and Drosophila human SOD in the motor neurons or the G85R mutation. We're seeing this failure of, with repeated stimulation, failure of muscle contraction, which you don't see when you drive just Drosophila in these neurons and then addiction control. So again, they're saying that neurons are not functioning correctly. The neurons that are expressing this new protein that are innervating these flight muscles are not functioning correctly upon stimulation. So they're seeing movement defects. They think they're seeing aggregation, although it wasn't as clear as in the worm model. And they're seeing synaptic defects at these flight muscles and brain extension muscles. So here they're looking now more closely at this protein accumulation in the motor neurons. So instead of doing that biochemistry, they're actually looking in the neurons. So here is just looking at GFP. Here's G85R. And here you can see they're looking at the buildup of kind of this GFP label, and they're labeling here with just a neuronal marker ELAB. And they say that they're seeing aggregates, not in all the neurons, but in some of the neurons. And it gets worse over time. Okay, so like the worm, they're seeing these neuronal aggregates and they express movement. And here is accumulation. This is the human SOD accumulation. Here's the wild type SOD. Here's A4B and here's G85R. So again, you're seeing it both with the wild type and the mutants with G85R being worse. And this is the dark brown is the more severe. And it gets worse over time. And here's what they call no aggregation, mild, moderate, and severe aggregation. So both with biochemistry, again, just like the worm, both with biochemistry and looking at these neurons, they're seeing protein aggregation when you overexpress the mutant forms. And then what they showed, which was different than in the worm model, they actually looked at whether you were getting a stress response in these neurons. And what they found, looking at just HSP70, that you were getting an increase in HSP70, but it wasn't in neurons. So here's ELAB again. This is a marker for neurons. Here's the SOD aggregates in the U2 motor neurons. And here you're seeing this heat shock response in these cells that aren't overlapping with the ELAB or with the SOD or with GFP. And so then they used a marker for glial cells, and they said that this heat shock response was actually in glial cells. So what they're saying is that when you drive the mutant protein in motor neurons, you're actually seeing a stress response in the neighboring glial cells. So this is a little bit different than from what Dr. Kaspar was talking about, how the astrocytes and microglia are affecting the motor neurons. Here they're saying that motor neurons that are expressing mutant protein are actually affecting the neighboring glial cells. So that's something to keep in mind. So just to summarize this, the slides are seeing motor dysfunction. You aren't seeing any decrease in survival of the animal. You see the failure of high-frequency synaptic transmission in those two muscle groups that they analyzed. You see neuronal aggregates, and then a non-cell autonomous stress response in glia. The caveat being that the model only expressed mutant SOD motor neurons, so it wasn't expressed endogenous, where endogenous SOD is expressed, which is in all cell types. And they did not see any evidence of neuronal cell death. Any questions about it? All right, so I'm going to go on to zebrafish. And again, if you look at the conservation of the amino acids, you can see it's even greater in zebrafish, which is a vertebrate. So there's a lot higher conservation, about fish about 71% identical, 81% similar. And again, 2-point mutations were made, G93R, or G85R and G93R, and then common mutations. So this is actually from my lab. So what we did was, instead of using a promoter just driving the mutant SOD in a certain cell type, we wanted to use the endogenous promoter. And again, this is because of things that you guys have been learning about, right? Even though you have a disease that's showing up in one cell type, doesn't mean that that's what's driving the disease. Right? It could be the expression of that mutant SOD in neighboring cells that are impinging upon the motor neurons. So we wanted to use the endogenous promoter. And in a lot of these animals, it's heterologous promoters often don't work, so that's the other thing to think about. You know, which promoters work the best in the model system that you're using. So we knew from better expression and using, say, in addition, because the zebrafish promoter hadn't been characterized, what we did was to make a back transgenic. So we took a large chunk of DNA and used about 11.7 kb upstream so it could encompass the whole promoter. We didn't make it much larger because it gets harder and harder to make transgenics if you have large pieces of DNA. So we took the back and kind of cut out the middle of it and used a large chunk of what we hoped would encompass the whole promoter, and then used the zebrafish SOD gene, where we cut out the middle and then made point mutations and then recombinated faster. So we then had a little tag here where we could have some fluorescence follow this transgene and then make transgenic animals. Looking at the copy number, we found a couple lines that had very high copy number expression by RT-PCR, but when we looked at the proteins, it was only about 3-fold overexpressed. So again, this is the other thing to think about when you're making these lines, is that sometimes you want high expression so you see the phenotype, but you also don't want such high expression that you see some aberrant phenotype. Actually, in this model, other people also tried to make these fish with different techniques, and it's very hard to make a fish overexpressing large amounts of SOD. The fish don't like it, they don't survive to breed. So this is actually the best line that we ever got, was expressing 3-fold more. So sometimes the animals limit you in ways that you can't really do anything about. People try really hard to get some expressing higher levels, and it's unsuccessful. So we overexpressed both. We made transgenic expressing both mutant SOD and then the same kind of transgenic expressing wild type SOD. So we had high copy number with wild type and then a lower copy number for protein level with wild type which as well. So just by looking, doing antibody labeling of the synapses, again looking at these presynaptic components that were looked at in other animals, we saw that there was a subtle change early on in the larvae, but progressively got worse. So by the time you got to adults, you were seeing that the synapses were very fragmented. So the synapses in zebrafish, just like in Drosophila, are actually puncta. They're not these pretzel-shaped synapses that you see in mammals. So you see these long kind of, first of all, you just see puncta, but then you see kind of these longer stripes of synapses here. And these were fragmented in humans. And that was more dramatic as time went on. So you saw a decrease in the presynaptic area and a loss of juxtaposition between pre and postsynaptic components of the synapse. So seeing synapse defects in these animals all along the trunk muscle. To look for movement, so the fish didn't have any just early on, no just normal movement behavior problems. We did circadian rhythm, looked at activity, and it didn't seem to be affected. So we wanted to actually test fish strength. So the one way we thought to do that was to do a swim tunnel. So we had this built for us, and what it is that you have water. You have water flowing in a current along a tube, and then you put the adult fish in there, and it swims. Fish normally just naturally swim against the current. So these fish will swim against the current, and you turn up the current, you see how long the fish can maintain it and let it fall back. Okay? And then from that, you could calculate the critical swimming speed. And what we found is if we looked at non-transgenic fish and wild type overexpressing fish, that they had a similar profile. But if we compare non-transgenic to the G93R, we saw that these had a decreased critical swimming speed. So it went from about 15 to about 9. And this is true of both of the lines that we made, the G93R and the G85R. Oh, just two different lines of G93R. So they both had decreased critical swimming speed. We see this phenotype starting at about 6 months of age, and by 10 months of age, they all seem to have this problem. So it's a very consistent movement effect. In addition, we looked at muscle physiology. This was working with Paul Jansen here. We were interested in asking whether the problems they were having with swimming and the swimming behavior was due to a problem with muscle contraction. So we found that there was no difference in the maximal twitch force in these animals, indicating that the muscle contractile properties were intact. better for the muscle. And these mutants don't lack that activity. So this could just be a kind of a byproduct of having more SOD in these animals. But certainly we weren't seeing a problem with the muscle physiology or the contractile forces, suggesting that the problems that we were seeing with the movement was due to the problems in the nervous system and not the problems in the muscle. Oh, that's a video that I although also end-stage fish. So these fish actually started having movement defects in the second half of the first year, and then a lot of them didn't die until like a year and a half. So this was a much longer-lived model. And at end stage, these animals showed decreased chat labeling in the spinal cord. And the reason that we call these fish end stage was that, I can show you the video, but the animals couldn't swim up in the water tank. They would lay at the bottom of the tank. They would kind of twitch around. So they had, by the time they died, they had serious movement defects. So they were, it's very rare to see a paralyzed fish, but these fish weren't moving well and weren't moving much on their own unless they were forced to move. So at that stage, we saw a decrease in motor neuron number. So this is really different than invertebrate models where there was no loss of motor neurons and no actual loss of decrease in lifespan. But again, this was also driving SOD in all cell types. This is just doing some EM and showing some of this pathology in the spinal cord and muscle. So we're seeing shrunken motor neurons, vacuolated mitochondria, and disintegrating crispy. So again, kind of hallmarks of degenerating neurons. In addition, we saw changes in the muscle, which wasn't in all muscle. If you look along the muscle of the animal, there were patches of muscle that looked completely normal, and then patches of muscle that had reduced myofibril size, smaller mitochondria, and collagen deposition, okay, and just a disorganization of the normal muscle bands. In addition, like I mentioned, we saw premature death in these animals. Not all of them, so there was some variability, but these G90PR, OS10 higher expressing lines, and also the G90PR medium expressing lines, both showed about half the animals or so dying. But again, there's variability, and this is something to think about when we're talking about animals that aren't inbred, right? So with the mouse models that you'll hear more about in another lecture, these animals are pretty consistent in when they get the disease and when they get to end stage. And this isn't true of zebrafish. There are some animals that die for like 3 years, and this could be due to the outbred nature of the animals. Good question.
Yeah, so is that one time and then mutant SOD1 time expressing the same copy number and then one?
Right, so this wild type OS1 is expressing the same copy number as this one here. So these two have the same protein level, I should say, of overexpression, about 2-fold. And then this one's a little more severe. It has about 3-fold. So just a summary of the zebrafish model. We saw presynaptic defects that were progressive over time. Movement defects in that swim tunnel, but no overt muscle defects, at least until end stage. Motor neuron cell loss and premature death. And importantly, we didn't see any aggregates in this model. We looked both by eye and labeling with SOD antibodies and also by Western blots. So this is a difference that we found that we did not see, aggregates. So I'm not going to go over the mouse model. There's a lot of them. Yeah.
Have you ever done the compound screening using your fish model?
Compound screening? So we would like to, but because of the variability in some of these phenotypes, it's very difficult. So I wouldn't want to do an antibody labeling. So I think the best way to do the screening is the swim tunnel. And we're actually talking to a drug company about doing that type of screening. But it has to be scaled up. Because for the swim tunnel, you do one fish at a time. It's not hard, but it's labor intensive. So I don't think you would ever do a large-scale screening, but you could do a targeted screening. I think that's effective. But the other issue is, when do you treat the animals, right? Because you start to do the swim tunnel, you start to see defects in swimming at about 6 months of age. So would you treat larvae? Would you treat throughout for 6 months? So these are the kind of questions that we're starting to think about more seriously. What I would days. They're seeing changes in the ER stress response, the EPR response. They're seeing increases in the proteins that, like BIF and CHOP, that go in and try to take care of this folded proteins. So the goal would be with this model is to find a readout that happens in the first week of larval development that you could then visualize with the fluorescent marker. Right? Say you saw an increase in a protein during the first week. You knock in GFP or RFP and then have an animal where in the mutants that protein, that GFP signal is increased in that first week of development. Use that as a screen. That would be really powerful. Then you can do it with a screen. So it's just a matter of identifying the early changes that happen. And maybe they aren't. Maybe that's not, maybe you won't find those changes. So that is one approach. Because then you use the strength of the zebrafish, right? Early larval screens is where you want to use zebrafish. They're off rack. They're 24 well plates, 96 well plates. You can do larger numbers. So that's kind of the goal. But right now we don't have an early readout. So this is something I've been interested in for years because we haven't found it. Really hard to find what it is. So that would be a really great way to do screening. Because then you have a whole animal model, right? So you're not using some fish. So this is just a quick summary of the mouse model. So many of the mouse models, there's a ton of them, right? But some of the first ones that were made, they do use the endogenous mouse promoter. That's actually why we did zebrafish the way we did. We looked at kind of the mouse model today. We really liked the way that was doing it because again, then you're driving expression in the right cell types. I think that's hugely important. Mostly we don't understand the disease. We usually don't. They did see neuronal aggregates in some of their models. Loss of motor neurons, decreased movement followed by paralysis, axon sprouting and reinnervation. This is not something that you see in zebrafish, and you don't see it in invertebrate models either. So this axon sprouting and reinnervation is more of a mammalian thing. So if that is a big component of the disease, then the mouse model is an important place to look for that. As was mentioned earlier, a commonly used model, the G93A mice has about 17 to 20-fold protein expression, which is really high. They're inbred lines, so again, the question of will small changes translate to outbred systems? And sick animals make drug and genetic screens very difficult for mouse. So again, doing large-scale drug screens is really not very feasible in these mouse models, at least not in this mouse model. So just things that I think you should think about when you use animal models to study disease. So some of the common things that were found in all these models, presynaptic components were affected, movement affected, right? The neuronal aggregates, I'm not really sure. I talked to Bob Brown, who's a big ALS researcher with his clinician. He says you don't always see neuronal aggregates in human patient material. So it's unclear what to make of those neuronal aggregates. Is it just a matter of how much protein is being expressed, drivers that you're using? Yeah, so does anybody study neuronal aggregates in the disease models? Has any strong opinions about these neuronal aggregates? I think it's pretty much up in the air. It could just be a byproduct of expressing the protein at high levels. But there are some common phenotypes. So I guess one thing to think about is when you see the same phenotype in 4 different animal models, does that make you feel good about that phenotype? So the fact that there's presynaptic defects, with movement defects. So there's clearly motor neuron dysfunction in all of these animals. Whether motor neurons die or not is not concerned. The invertebrate animals, the motor neurons don't die, but that could be a time-based problem. But certainly all of them have motor deficits. So, that's a common theme that came out of all the different animal model studies. I think it's clear that using those invertebrate screens can be important, but then it has to be translated, put into a model where the protein is being expressed in all the relevant cell types, not just the motor neurons. And again, is it relevant to express mutant proteins only in selected cell types? Is that a good way to build a model? Should read a lot of caveats, mostly from what we've learned about ALS. It's not just the motor neuron. The strength of the non-mammalian models for genetics and drug screens is strong. think about this on a daily basis? What's the thought in the field nowadays about that? Is it still hotly debated?
Yeah, I mean,
Yeah. Yeah.
that's again, I would say, is a problem because the models we have are very limited. As you said, we have a couple of sort of one mouse models, they are great, but that's two mutations, three mutations. Clearly there, as Brian showed last week, I mean, the studies from the Cleveland lab as well as other labs would indicate that the motor neurons are affected pretty early and might then, probably activate over cell types. There's also studies from other labs showing ER stress as early as, I think, even 15 days postnatally or 30 days postnatally. So really early, the neurons are stressed, although they're still there, they're alive, they're functioning. So it's happening early. So I would say that from the models, you could probably say the motor neurons are likely involved in onset in the animal models. I think that's pretty common thought. But how much? It's also the most